Great, thanks for the introduction, Steph. Thanks everyone for being here. I'm excited to talk a little bit about research I've done in the past and sort of where my research is going in the next couple of years. Hopefully I'll get through all this in 7 minutes, but I'm really excited to start the conversation as well. Most of my research focuses on these two questions, sort of how different could life be? It's a central question in synthetic biology as we start to rearrange and remix the components of biological systems. It's also a central question in evolutionary biology about the origin of life, about what's contingent and what's necessary in biology as we see it today. When you engage with the idea that life could be very different as we see it, that it could use different functional polymers or different information storage, you're left with this question of would we even recognize it if we saw it? That might seem like a sort of lofty, abstract question that you think about when you drink whiskey or wine, but it's a practical question for people at NASA that are sending missions within the solar system and planning manned missions in the coming decades. So one way that we try to approach this problem, where what we want to do is detect life without knowing what the biochemistry is. We want to look for something without specifying what that thing precisely is. The way we try to approach this is the framework that we call assembly theory. This is based on the hypothesis that living systems are unique insofar as they can make complex molecules or complex objects. So when I say living systems, I include microbes or proteins, but I also include chemists and scientists in the lab. So this measure of complexity that we came up with, we call the assembly index or the molecular assembly index. It's based on the sort of property of molecular graphs with a small picture there. I won't go into the details of the calculation. I'll just show some examples so you can get some intuition about what's happening here. So the assembly index, the MA here, of some different molecules is shown. Some of these we find in meteorites, like glycine, serine, simple amino acids. They're central in biology, but we know they can form abiotically. Meanwhile, when you get to the bottom right here, we're looking at things that are sort of natural products or more complex synthetic compounds that we don't expect to form, quote unquote, on accident without either a cell or a scientist trying to steer their construction. One thing that's different about this measure of complexity compared to other measures of complexity that have been proposed in computer science and in physics is that we can measure this in the lab. So even though when we do this calculation, we don't worry too much about the law of physics or chemistry, when we get the end result, it's something that we can observe. In this case, I'm going to show some data where we use tandem mass spectrometry, sort of the workhorse of proteomics and metabolomics. You can do this with NMR or IR as well. I'll focus on mass spec because there's a history of studying these in space. The intuition is more complex molecules produce more distinct ions when you fragment them. It's not that complicated. There's some examples here. Here's a really complicated molecule. When you hit this hard and break it into pieces, it produces more pieces. Simple molecule, simple pieces. This is what this looks like if you do this over a bunch of small organics and peptides. There's a lot of variation here, but what this means is that if we can fragment the molecule in our mass spec, we can count up the number of unique ions it generates and get a good guess at what the complexity is. Same intuition works for NMR and IR. So we took this and we said, okay, let's apply this to some more complex samples. So we went to the lab and we gathered up a bunch of stuff that we could make or analyze. So here's the results of a bunch of different mass spec experiments. The vertical axis here is a bunch of different samples. So we've got things that we made, things that we found, some blinded samples that were sent to us. The horizontal axis is the measure of complexity. The sort of bold dots that you see are the most complex ion we saw in the sample, and the smaller faded ones are other ions from the same sample, but were less complex. The different colors correspond to different types of samples. The red ones were abiotic controls, basically prebiotic chemistry experiments. The green ones are the biological samples. And then there's a few other controls that we did, but the most interesting ones were these blinded samples that were sent to us from our collaborators at NASA. We didn't know what these were when we put them in the instrument. The main takeaway is that only the biological samples produced results that were farther on this end of the spectrum. So what this means is that we can deploy this as a means for searching for life, even if we don't specify the biochemistry exactly. There's more work to do. This is a hypothesis. This is only some data, not all possible data. But it gives us a way to start to take this to other worlds and start to imagine these experiments going forward. So this is great, but as soon as you start thinking about doing any kind of analytical chemistry in space, you realize you're confronted with these very extreme conditions. And the ultimate goal is going to be, how much can we learn about this planet or this biochemistry with very limited samples, either because your instrument can only be used so many times or because you only have so much power, you're very far away from Earth. And so an aspect of my research now is sort of trying to use computation and these physics-informed models to boost our ability to do inference, where we have very limited access to the chemistry we're interested in. So here's a model where I'm working with some collaborators on doing some surface calculus inside a rock, right? We're flowing some compounds through a rock and trying to make some cofactors. And we don't have direct access to the interior, and so the question is, how good can we get the reaction kinetics, even if we don't have perfect information? And it seems like we can get away with very little as long as we do the inference not with like a giant neural network with billions of parameters, but we take a guess at what the actual underlying physics is, and we fit directly to that. Just one other thing that I'll mention here is, as soon as you start thinking about alternative biochemistries, alternative functional polymers, you might ask, well, how would you sequence oligomers if you found them somewhere else? It might be interesting for somebody like me who thinks about prebiotic chemistry. This might also be useful if you're putting non-conventional amino acids into proteins, or if you're using synthetic polymers and trying to program the exact sequence of those monomers in your oligomer. I worked on a project where we started from the monomers and showed that you could use tandem aspect to sequence any oligomers, whether they're biologically derived or modified, prebiotic, or even synthetic. One interesting case, if we use it to sequence nucleic acids, you might wonder, why would you use tandem aspect to sequence nucleic acids? Well, one advantage is we could get the sequence as well as the methylation site. So we ran a test where we put the exact same sequence with a methylation site of the backbone in two different places, and using this measurement and this tool, we could predict exactly where the methylation site was with high confidence. There's more I'd love to talk about, but I'll stop there for today. Here's my email and my office. I'm happy to take questions now, but also just knock at my door, love to chat and start new conversations. Thanks.